Microbiology explores the incredible diversity of life at the microscopic level. In this lecture, we'll explore how these organisms are organized into three broad categories: the eukaryotic organisms, the prokaryotic organisms, and acellular entities. After this lecture, it's my goal that you'll not only be able to recognize the differences between these groups, but you'll also see why some will stretch the limits of what we consider as being living organisms. In the Introduction to Microbiology lecture, I mentioned that the cell theory states three things. All living organisms are made of one or more cells. This means that the cells are the building blocks of life. Second, the cell is the basic unit of structure and function in living cells. It carries out all the processes that it needs for life. And thirdly, all cells come from pre-existing cells. Remember the theory of biogenesis. So new cells are formed by the division of existing cells, not spontaneously generated as previously thought. So putting these three statements together, the cell defines what we recognize as living organisms. For living organisms to grow and replicate, their cells need the following. First is the cell membrane, also called the plasma membrane. It's the boundary that separates the inside of the cell from its environment, and it also controls what enters and exits the cell. The cell membrane is thin. It's flexible barrier. It separates the internal environment from the external. It's primarily made of phospholipid bilayer. You'll also see, though, as an exception, is that the archaea will have a monolayer of phospholipids. But nonetheless, the primary molecule is phospholipids. Within the phospholipids, there will be proteins that are embedded, and this is going to give the membrane its selective permeability. In eukaryotic organisms and many bacterial cells, sterols, such as cholesterol or hopanoids, will also be embedded to help give the membrane its fluidity. These phospholipids need to move around. Overall, the cell membrane is going to act as a protective barrier in all the organisms. It controls the movement of substance in and out of the cell, allowing nutrients in, removing waste out, and regulating ions and molecules. In addition to this transport, it's essential for cell signaling, for recognizing foreign objects and interactions with its environment. It's going to make it vital for the survival and function of every cell. Secondly, the cell also needs genetic material, and this is in the form of both DNA and RNA. This genetic material are instructions for building proteins and for passing information along to the next generation. DNA in eukaryotic cells are found inside the nucleus, and they're often organized as multiple linear chromosomes. They're double-stranded, antiparallel, helical, but the key word here is that they are linear. Additional DNA are found in small amounts in organelles such as the mitochondria and chloroplasts for plants and algae, where it's usually circular, not linear, and resembles that of prokaryotic DNA. This takes us to the DNA in prokaryotes, where it's found in the cytoplasm, specifically in a region that's called the nucleoid. So prokaryotic cells do not have a nucleus. They have a nucleoid. So unlike that of the eukaryotic DNA, prokaryotic DNA is singular and it's circular. It's also double-stranded antiparallel, but there's only one, and it is attached at each end. They're not associated with histones, except for the archaea. Additional DNA might be contained in small amounts, and would also be circular DNA. These would be called plasmids, which carry extra genes. We'll talk more about these when we discuss the bacteria and archaea in a separate lecture. Next, the cell needs to contain a cytoplasm. This is the internal fluid. It's composed mostly of water with dissolved molecules, where metabolic reactions take place. In eukaryotic cells, it refers specifically to the region between the cell membrane and the nuclear envelope. The fluid that makes the cytoplasm is about 70 to 80% water, and it's mixed in with these soluble ions, small molecules, and macromolecules. In eukaryotic cells, it also contains these membrane-bound organelles that are suspended and organized with the aid of cytoskeletons. The cytoplasm is highly metabolically active, as mentioned, and it serves the site where many essential biochemical reactions for life will take place. Finally, all cells have ribosomes. They are non-membrane-bound structures that assemble proteins according to the cell's genetic information. These ribosomes help bring together messenger RNA and transfer RNA that's linked to their appropriate amino acids. Together, they help make proteins. I'd like you to pay attention to 70S versus 80S. The S stands for Svedberg rates, and it's a measurement of sedimentation during centrifugation, and it selects for size. For our purposes, 70S ribosomes are smaller than 80S ribosomes. Now, prokaryotic cells have the smaller version. They have the 70S ribosomes in their cytoplasm, whereas eukaryotic cells contain both 70S and 80S ribosomes. Their 70S ribosomes are found in the mitochondria and the chloroplasts, but their larger ones are found in the cytoplasm and attached to the surface of their rough endoplasmic reticulum. We'll see how these variations of these components among living organisms will help us classify them. So understanding what defines a cell and the principles of the cell theory helps us study and classify organisms as being eukaryotic or prokaryotic or acellular. The major differences between eukaryotic and prokaryotic are as follows. The eukaryotic cells contain a nucleus with a large linear chromosomes that are enclosed around by a nuclear envelope. They have multiple membrane-bound organelles, such as mitochondria and in the endoplasmic reticulum. Whereas the prokaryotic organisms, they lack this nucleus. Their DNA is usually a single circular chromosome that is located in a DNA-rich area in the cytoplasm called the nucleoid, and they don't have membrane-bound organelles. The acellular entities are not true cells, and they require a host to replicate them. These acellular entities lack the basic structures of living cells. They don't have a cell membrane, they don't have a cytoplasm, nor do they have ribosomes. Some may have either DNA or RNA, or no genetic makeup at all. They're unable to carry out independent metabolism, and they lack the ability to self-reproduce themselves. They need to rely on the host cell to help replicate them. So having defined what a eukaryotic organism and a prokaryotic organism are, as well as the acellular entities, we can now explore how microorganisms are categorized within these three groups. These microorganisms, or also known as microbes, are generally classified into one of the following categories. You have the protozoa, the algae, the fungi, the bacteria, the archaea, the viruses, the viroids, and prions. Let's start with the eukaryotic organisms. The protozoa, algae, and fungi are eukaryotic microbes. They might be singular cell, sometimes called unicellular, or they can also be multicellular organisms, but their cells all have a nucleus and membrane-bound organelles. As the semester progresses, you'll learn some major structural differences between these eukaryotic cells that will help differentiate one organism from another. But for this lecture, we'll start with a brief introduction to each type, starting with the protozoa. Protozoas are single-celled microorganisms. You will not find a multicellular protozoa. They lack a cell wall. Don't mix up cell wall with cell membrane. They typically are motile. They have the ability to move around in their environment at some stage in their life cycle. Some of them are going to be free-living and can be found in a variety of moist environments such as the soil, fresh, aquatic, or marine waters, while others are going to be parasitic in nature. They're going to live inside other organisms. They primarily generate energy through aerobic cellular respiration. That means that they break down organic matter to make their energy in the form of ATP. There are some exceptions, and one of them is that the protozoa euglena contains chloroplasts with chlorophyll in it, so that enables them to capture sunlight and convert it into chemical energy through the process of photosynthesis. But for general purposes, all protozoa use aerobic cellular respiration. This takes us to the next, the algae. They are also a diverse group of photosynthetic microorganisms. So just like plants, they have chloroplasts. They can do photosynthesis. These microorganisms can be single-celled, just like protozoa, but they can also be multicellular. If they're single-cell, they can sometimes live in a colony, in which these are basically single-cell organisms that are living together as a colony, called colonial, and they group together for their common good, such as gathering food or for reproduction. If they're multicellular, they could be filamentous. That means cells are end-to-end, or they could be these large, complex, multicellular organisms, such as shown here as in the form of kelp. Their sizes can range from microscopic, as shown here on the left, and they can be macroscopic, such as kelp. No matter what, they all contain chlorophyll, and they can contain other light-absorbing pigments that help them capture light and energy to produce food by photosynthesis. Algae will display a variety of colors because they all have chlorophyll that would keep them as green, but because they have a combination of other light-absorbing pigments, that will give them different colors. These pigments will absorb light at different wavelengths, which will allow them to capture energy efficiently in a wide range of aquatic environments. For instance, some might be on the surface of an aquatic environment, whereas others can go deeper depending on the light pigments and the light that can be absorbed by those pigments. Again, we'll talk more about this when we talk about algae. So for this purpose, I'd like you to know that algae are photosynthetic, and they have different light-activating pigments. Besides chlorophyll, they'll have others that give them different colors. This takes us to the last microorganisms that are eukaryotic, and these are the fungi. Just like the protozoa and algae, the fungi are a diverse group of organisms. They mainly obtain their nutrients by secreting exoenzymes. So exoenzymes is just a fancy name to say that these enzymes are released outside, hence the name, the prefix exo. These enzymes are released outside the cell, where they can break down large organic material that's outside in the environment, and then they can absorb those smaller molecules back inside the cell. So the resulting smaller molecules are absorbed across the cell membrane and further metabolized through aerobic cellular respiration to produce energy in the form of ATP. These fungi can be single-cell, like shown here as in the form of yeast, or they can be multicellular, such as shown here as mold, or mushrooms, which is not shown here. That takes us to the next microorganisms, the bacteria and the archaea, which are formed by prokaryotic cells. The prokaryotic, because their cells do not have a membrane-bound nucleus, nor do they have membrane-bound organelles. So let's talk about the bacteria first. Bacteria are single-celled prokaryotic organisms. They are all single-celled. You will not find a multicellular prokaryotic organism. This goes for the bacteria and the archaea. Because they don't have membrane-bound organelles, they're much simpler in their cellular structure than the eukaryotic organisms, and they are significantly smaller. Some of these bacteria can live freely in the environment, while others are parasitic. They have a rigid outer cell wall. This cell wall protects them in aqueous environments and gives them a characteristic shape. So when we examine bacteria under the microscope, we'll see that they have different shapes. And I'm showing you some in the picture here. Showing you here on the left, they are long rod shaped. We call them bacillus shaped. These here in the middle are more circular and they're clustered together. But if you look individually, they're little dots. These dots are coccus. And then you might have these long corkscrew shaped are called spirillum. So these shapes are unique and they're based on their cell wall formation. Next are the archaea. They have similar structure to that of bacteria, but they also have differences. Their differences would be in their cell wall and their cell membrane. Also, their genetic makeup is a little bit different, and they have different biochemical traits that help distinguish them between being archaea and bacteria. That leaves us to the last group, the acellular entities. These are the viruses, the viroids, and the prions. Recall that acellular entities lack the basic cell structures of living cells. They don't have a cell membrane, they don't have a cytoplasm, nor do they have ribosomes. Let's start by talking about the viruses. These viruses do have genetic makeup, but unlike the eukaryotic or the prokaryotic cells, they can either have DNA or RNA. They'll never have both. This is going to help us categorize them as either being DNA viruses or RNA viruses. In either case, their genetic material, their DNA or their RNA, will be surrounded for protection by a protein capsid. It's basically a coating around that is made out of proteins. Some viruses might have an extra molecule called a lipid envelope. So we'll talk about whether they're enveloped or non-enveloped. All viruses are parasitic. They're unable to carry out metabolism by themselves, and they rely solely on the host cell to produce more virus particles. Next are the viroids. Try not to confuse viruses and viroids. Viroids only have RNA as a genetic makeup. The only thing that they are is just a small circular RNA molecule that infects plants. So they are plant parasites. And unlike viruses, they don't have a protein coating around them. So it's just that genetic information, that RNA. This makes them much more simpler than viruses, and they are entirely parasitic, and they rely solely on the plant host cell, to help replicate more RNA molecules. Last are the prions. And these don't have any genetic makeup at all. They don't have RNA or DNA. They're composed purely of a small piece of protein. And as simple as they are, they cause neurodegenerative diseases. You must have heard of mad cow disease. Prions are the cause of mad cow disease. We're going to talk about other human diseases that are caused by prions in the semester. Prions are simply a misfolded form of proteins. they can force normal proteins in the host to misfold, leading to the progression of the disease. They don't contain any genetic material, and they rely solely on this protein-based mechanism to replicate more misfolded proteins. I mentioned about the prokaryotes being about 10 to 100 times smaller. So I'd like to just step away for a minute and talk about these sizes of microorganisms and show you them on a chart. So eukaryotic cells are generally the largest. And they're shown right here between 100 and 1. So they're typically ranging between 10 and 100 micrometers in diameter. They have different shapes depending on the type of cells that they are. Whereas the prokaryotic organisms, that's these guys right here, they're much smaller, and they're usually around between 0.1 to 5 micrometers or micrometers. It takes a light microscope to be able to see these prokaryotic organisms under a magnification of 1,000 times. Lastly are the acellular entities, such as the viruses. So here's the viruses right here. Notice that we go from micrometer scale to nanometer scales. So viruses, viroids, and prions. Remember, prions are just a small protein. They're much smaller than the prokaryotic organisms, and they're measured as nanometers. Therefore, a light microscope cannot detect them. It's going to require the use of an electron microscope in order to see these guys. Let's see how they are related to each other. In the Introduction to Microbiology lecture, I mentioned the tree of life. Remember, this was designed by Carl Woese. This diagram places all known living organisms along an evolutionary timeline based on their genetic similarities with their ribosomal RNA sequences. Each of these organisms is represented at the tips of the branches. So here's the tips of the branches, the outermost part of the tree. While the length of each branch represents the evolutionary time since they diverged from their ancestral lineage. So for instance, here's the length of a tree, and this where the thermotoga emerged or diverged from this ancestor right here. And then they further branched off to form the green filamentous bacteria and this cluster here. So points where the branches meet represents their common ancestor. These phylogenetic trees, like this tree of life, help us trace shared ancestry and compare how recent different groups split from each other. Because this tree is built from ribosomal RNA sequences, acellular entities such as the viruses, prions, and viroids are not included in this tree because they don't have ribosomes. So the base of this tree, down here, represents the common ancestor that links all living organisms. It's believed to have lived 3.5 billion years ago as a single-cell prokaryotic organism. Soon after that, about 1 billion years later, it's believed that the prokaryotic lineage split into 2 branches, representing the bacteria and the archaea. About 1.5 billion years after that, the first eukaryotes split apart from the archaea lineage. Remember, we mentioned the endosymbiotic theory. That explains how these eukaryotic organisms formed from a prokaryotic ancestor by accounting for their formation of mitochondria and chloroplasts. So this is the slide that represents the endosymbiotic theory. The theory states that a large ancestral eukaryotic cell engulfed but did not digest a small prokaryotic cell that was capable of aerobic respiration. So here's our ancestral prokaryotic organisms. He got a little larger, and notice that the membrane is starting to internalize. That's going to lead to other membrane-bound organelles. But here's that small prokaryotic organisms in orange that was engulfed by the larger prokaryotic organism. He engulfed him, and he stayed inside, and he eventually became the mitochondria. Likewise, some ancestral cells may also have engulfed small prokaryotic organisms, such as this one, that were able to undergo photosynthesis. These stayed within the larger prokaryotic organism and later became the chloroplasts. So over time, these engulfed small prokaryotic cells became permanent organelles. The ancestral eukaryotic cell internalized its membranes to form the endoplasmic reticulum, the Golgi bodies, and other organelles. And eventually, this cell became a eukaryotic organism. The evidence that supports this is that the mitochondria and the chloroplasts have their own circular DNA, similar to that of prokaryotic organisms. And they also replicate similar to that of bacteria, and that is by binary fission. We'll talk more about binary fission as we go on. And the genetic information that they have similarities to that of bacteria, some prokaryotic organisms. So this is the theory of how a eukaryotic organism branched off from the lineage of a prokaryotic organism. So let's come back to this slide just to make another point. I've defined what a eukaryotic cell is and what a prokaryotic cell is, and I defined what differentiates one from the other. I also said that microbes, also known as microorganisms, are living organisms. So when you hear the word organism, you assume it's something that's living. Because they're micro, they're too small to be seen without the use of a microscope. These include the protozoas, all the way down to the prions. But which one of these microbes are actually living or alive? All of them? None of them? Some of them? If not all, which ones are not living and which ones are living? And how do we decide which one's living and not? So I'd like you to think about this. Acellular microbes challenge this traditional definition of life since they can't reproduce or metabolize on their own, but they can direct host cells to reproduce new infectious particles. So should they be considered as microbes? Remember, microbes, microorganisms, organisms are living. By this traditional definition of microorganisms, the answer would be yes. They should be considered as living. But by a strict biological definition, the answer is actually no, because they lack these essential cellular machinery that's needed for independent replication. Remember, viruses rely on a host cell to make more virus copies. Prions rely on a misshapen prion protein to force other prion proteins to misfold. And viroids are just a circular-shaped RNA molecule that relies on the plant host cell to make more copies of this circular RNA molecule. So, are they living or are they not? I believe that they are not. So I would not call these virus cells, prion cells, or viroid cells. I would call them virus particles, prion proteins, and viroid RNA. So hence they are acellular entities. I'm including this image in your notes that will help you review the material that was presented so that you can study this for your exams. Likewise, this slide summarizes some of the concepts that were presented in this lecture. So in summary, I hope that you've can appreciate that life on Earth exists in a remarkable diversity of forms. The prokaryotic microbes, for example, like bacteria and archaea, are simple. They're single-cell organisms without a nucleus, yet they can thrive in virtually every environment. We're going to show you that they are the most diverse organisms present on Earth. Eukaryotic microbes, such as the fungi, the algae, and the protozoa, are more complex. They also have membrane-bound organelles, and they have specialized functions. And then there's these acellular entities, like the viruses and the viroids and the prions, which challenge our traditional definitions of life because they don't reproduce themselves. They're metabolic. They cannot metabolize on their own, but they can manipulate a host cell to propagate. We'll discuss these microbes in more detail as the semester progresses, and I hope that you'll learn how to differentiate one from the other based on their structure and their life cycles. Understanding these distinctions will help you appreciate the incredible variations or variety of microbe life and their profound impact, not only on us and our health, but on our ecosystem and for their use in biotechnology. So with that, this concludes our lecture on eukaryotic organisms, prokaryotic organisms, and acellular entities.